Hi, my name is Willy. Today I would like to introduce a mystery animal called Elisha viridis. It is a kind of sea slug that lives in the shallow water. In appearance, it looks like a little leaf, and actually, they can do exactly what it can do. First, remember back to primary school biology. We know that chloroplast is a characteristic of a plant, right? Animals don't have chloroplast. Animals don't have chlorophyll. Animals can't photosynthesis. But all has been a past. These guys, they feed on algae such as Codium tomentosum to take out the chloroplast. During digestion, the photosynthesis cells from the algae are only partially destroyed. Their chloroplasts are remain complete and allow Elisha viridis to take advantage of it. They are able to keep them from a long periods of time, and if you shine light on the animal, it photosynthesis. It fixes carbon and blows up oxygen. It is an example of chloroplastic symbiosis or calotoplasty. In other words, it's chloroplast robbery. Now, the question is, how can it do that? Unfortunately, as yet, nobody clearly knows how they keep their chloroplast running. But there are a couple of theories told that they proceed for dental gene transfer to turn the genes in the algae into its own. And the other thing is that the chloroplast gene already exists in its body since their birth. Although this is hard to make a conclusion on how they use it, but first of all, we also can realize on how important they use it. So in this research, calotoplasty photosynthesis is nutritionally relevant in the sea slug. We will focus on the importance of photosynthesis to the life of Elisha viridis. The calotoplasty in Chinese is called 稻链体. It is a special chloroplast that can run in animal's body. For this purpose, they present three hypothetical questions. First, if there is no lighting, no eating, what is the body influence of Elisha viridis? And next, the function of calotoplasty in the darkness. And last, how is calotoplasty abundance affected by starvation and light dark conditions? The methods are samples of sea slugs and algae were collected in the intertidal rocky zone of the Bruges Beach in Portugal. They took 30 samples of sea slugs at first. After they were brought into the laboratory, they were maintained in the seawater with a fixed temperature and salinity, plus printed in light for 16 days to allow them adapt in the laboratory environment. After that, pick up 3 of these 30 sea slugs randomly and divided them into control group and treatment group. The basis of distinction is their different lighting conditions. The experiment time is 24 days and measured in the day 3, 10, 17, and 24 in units of 7 days. The first one, slug A is normal lighting and normal eating as a control group. And treatment group, slug B is no eating but discontinuous lights about 14 hours light and 10 hours darkness in the day. And last, slug C is no lighting and no eating, put in the space of total starvation and darkness. Then we are able to observe what's the difference between these sea slugs. The experiment conclusion is that we can find that slug B is heavier than slug C significantly, and both of the treatment groups were lower than the control group. Their health is clearly reflected in their weight. Then go on the next diagram. Then we can also observe that slug C has gradually become shorter over time, but there is no significant difference of slug B. It is still close to the control group. So we check out the hyperspectral imaging. In this picture, the more red part, the higher density of calotoplasty, and the blue part quite opposite. we can draw two conclusions from this picture. First one, we can find that the density of chelotoplasts are highly distributed in their parapodia, right there. Then focusing on soft beak, although it maintains its body length, but its red pod and parapodia are almost disappeared. Not to mention Sloxy, it shrinks dramatically. So scientists speculated that they threw breakdown of chelotoplast to satisfy its energy requirement. That is to say, they turn chelotoplast into energy. So that's the reason they look thinner and eventually shrink. There's other evidence that can prove this. The density of chlorophyll A. We can find that both of the treatment groups' density were decreased and the SWAT C has greatly declined. So their ability of photosynthesis has become weak and eventually they will lose this ability. And the other conclusion is that even in a state of total starvation, Anisha viridis can also do photosynthesis to retain a certain limit of living energy. So after having this data, we can back to the first three questions. First, body influence. According to the first two diagrams, we know that they will be thinner first and shorter because their chelotoplast is highly distributed in their parapodia, and they cannot obtain energy from photosynthesis, so they threw breakdown of chelotoplast to supply its latent energy. And next, are chelotoplasts keep working in the darkness? No, just like normal chloroplasts, they are unable to photosynthesis in the darkness. And last, abundance. In the case of total starvation, as long as there is still a little light that can photosynthesis, Anisha viridis can also still retain a certain abundance of chelotoplast and body length. So, according to this research, we can realize the importance of photosynthesis to Anisha viridis, and can even say this is part of their life. There's even data from other research showing that they do not need to obtain external energy from up to 1 year. That is to say, they can live for 1 year without eating anything. So remember this conclusion that's digressed a little bit in the remaining time. This animal is called zebrafish. They are researched by scientists in the biomedical area actively because of their strong ability to regenerate. And scientists hope to transfer their genes to humans to cure some genetic disease like Parkinson's. Person gets Parkinson's when the brain cells that produce dopamine start to disappear. But zebrafish has the ability to regenerate cells on command. So if we can somehow give this ability to humans, they are germinal cells. Likewise, what if we can give the ability to photosynthesis to humans? What can we solve? The food crisis is not a problem anymore, right? Because your nutrition source is yourself instead of hamburger. So the only things you have to do in your life is lie on the bed and do nothing, sometimes back in the sun, then you can survive. It's very attractive if you can look at that you are turning green. Sounds a little bit crazy, but gene transfer is exactly what scientists are researching on. So how can you assert that this is impossible? That's all my presentation. Thanks for listening.
I think he had very good stage performance and talk speed, and his artwork is very good. But I think some difficult terms like kleptoplasty, I think he need to explain in his word instead of printing Chinese translation on it.
I think you have a great stage performer. You always looking at audience, but you speak too fast that I got a little bit hard to follow.